let's move to the last part of my seminars, the one on environmental control of schistosomiasis and specifically biological control. And see again how integration of data and modeling can help us to understand what are the potential benefits and also the drawback of using certain type of environmental control. What type of environmental control in the first place? I mentioned that in the first section of my talk. Well, the most important one is certainly this is called WASH, the water sanitation and hygiene. This is a crucial component of integrated schistosomiasis control. So it's certainly of major importance. It has some drawbacks though, or some challenges. The WASH interventions take a lot of time to develop. There are infrastructural interventions in a lot of cases. They have a significant upfront cost of investment. And they also might require significant behavioral changes in order to become part of the lifestyles of the places where we want to deploy. There is another alternative that is just to use molluscicide, just try to kill the snails where they are. And using molluscicide, yes, it's effective and control the snails, at least in the short term, but it has also negative environmental impact. And in addition, as we know, is the use of any insecticides and molluscicide are not that different, might eventually lead to selection of resistance. So relying entirely on molluscicide is a challenge, and it's difficult to be socially accepted in the places where there is a lot of transmissions. It doesn't really work. The question we want to address here in the third part of the seminar is whether biological control has a role in schistosomiasis reduction and elimination. Integrated pest management include biological control, and it's something that as humankind we have been done basically for centuries, almost a couple of millennia. There are examples that dates back to 1700s years ago. Biological control was first used more than a century ago at this point. And if you're Googling on biological control, you'll find 10 millions of instances. So it's, yes, very well established, but there is much less evidence of effectiveness of biological control for human diseases. We are living a new renaissance of phage therapy. There's been a lot of discussion in the case of mosquitoes. We know that might work in the lab conceptually, but there are very few instances in which this has been shown to be effective also, so to speak, in the real world. And what about schistosomiasis? Evidence of biological control typically emerge from extirpation of natural predators. So, and I want to show you the case here again on Senegal. You remember in the first part of my seminar, I showed that the construction of the Jama Dam has dramatically changed the landscape, creating better habitat for the snail to thrive. Now, there is something that you can't see from the satellite imagery, and this is the Macrobrachium volanovi, that is the African river prawn. And this prawn is native, as I was mentioning, is a voracious predator of the snails that amplify schisto. And I know you would ask is, no, it does not get infected when it eats a snail that is infected. This freshwater prawns is actually a migratory species. It requires brackish water for reproductions, where the larvae can develop in the first few weeks, and eventually then the prawns swim back, and it can cross up to 1,000 kilometers, typically several hundred kilometers upstream, and then go back for reproduction. Well, guess what? Building on a dam basically blocked the migratory patterns and prevented the prawns to basically complete their life cycles as going upstream. And with no predators, we remove an important agent of top-down control of the snail population, and this, in addition, with better habitat, led to this explosion of snails. So one of the things that we have been investigating in the past year is whether the control of disease transmission can be improved by restoring also the lost knowledge predator. So basically, whether we can add or complement the drug administration prosequental by restoring the prawns so that we can try to target both arms of transmission. And what we did, we've been working a lot, first in laboratory experiments, and Sokolov, Schwartz has been in summer interns here, and with controlled trial field experiment that were conducted in 2 phases. The first phase is between 2012 and 2013, and then other phases between 2015 and 2018, and also a series of modeling analysis. On a paper that Anna Sokolov published in Acta Tropica in 2014, show basically a relationship between prawn weight and the biomass of snails that are consumed by a prawn per day, which is one of the pillars of the so-called functional response of how many snails per unit time a prawn is able to eat, given the prawn size and the snail size. And we also saw that, for instance, that snails that are infected have a slower anti-predatory response, and so they can be predated at a higher rate, which is actually good. In first phase between 2012 and 2013, we show that stocking prawns has been able actually to reduce the abundance of Bulinus truncatus for schistosoma hematobians. So prawns were able, in fact, to control the snail, and the risk of schistosomiasis reinfection in school-age children. So, when we look at the human side, was also reduced thanks to restocking prawns in the places where they've been eliminated. Now, these results and this effect of predation to snail abundance, it kind of dismissed in the analysis of schistosomiasis control. Part of the reason, I think, is that historically, when we go back a lot, like in the 80s and in the 90s, a series of models and analysis, which were super sophisticated, actually, so really nice, were based on an old McDonald Ross type of model that basically track yes, the mean parasites burden, so the mean number of parasites per people, but just the prevalence of infection in snails, not the abundance in snails. And so the snail number was basically constant. So we basically took these models and started to say, well, explicitly track the number of infected snails, and then allow snail reproduction and replenishment and variable population size. Explicitly track the long latency time, so compartmental models of snail infections on the snail side that compute also the snails that were infected but not infectious yet. And last but not least, we account for prawn stocking and in a way that we could. And then we basically also use the reinfection data that we had with a maximum likelihood estimator to estimate transmission rate from snail to people and from eggs to snails. Those are parameters that are otherwise almost impossible to basically estimate empirically through direct observations. When we put that all together, so we look at the long-term equilibrium predictions, where we get this prawn abundance over 200 square meters, so how many prawns we had to stock, and we use this to estimate the number of infected snails at long-term equilibrium, the total snail abundance, the mean parasites burdening people, and the prevalence in human population, prevalence of infection in the human population. And what we see is that there is a prawn abundance, so an abundance of the predator or a prawn density that actually allow to remove entirely the infection in the long run, although the snail population is not wiped, but is reduced to a level that in life expectancy of the snails is reduced to a level that it does not allow for transmission any longer. And in fact, the mean parasite burden would drop down to very low level, although the prevalence in human infection dropped down a little bit less because there's going to be, fewer parasites still in a fairly wide range of the population. But these basically show that, at least from the modeling point of view, is potentially we can reach the density of predators that allow for the elimination of the disease. There are additional considerations, of course. If we do only mass drug administration, if we treat only people with praziquantel and repeat this year after year for 10 years, we can show that we can reduce the number of infected snails, because the parasite is a castrator. By reducing the number of infected snails, we actually increased the snail, because we're going to have more snails that are reproducing when we have fewer parasites. The number of worm per person, the mean parasite burden, decreases, and prevalence decreases as well. But if for any reason we stop administering praziquantel, because COVID-19 prevent to do that, then the snail, the worm abundance bounce back very rapidly. Snail bounce back, worm abundance bounce back, and disease go back quickly to the same level. If we only stock prawns on the other end, we could achieve pretty rapidly a density of snails to level that do not allow transmission from snail to parasite to humans any longer. But as you can see here, the prevalence of infection and the mean parasite burden take years to decrease. And the reason is that the parasite is really long living in the human blood vessels. They can stay there up to 30 years and on an average between 5 to 10 years. So it's very long to eliminate. If we complement mass drug administration with prawn intervention, then we can not only reduce the infected snails very quickly, and but also in few years, we could potentially achieve elimination of schistosomiasis. So integrated medical and biological control is the most effective strategy for schistosomiasis, where when I say integrated, we can use biological control, and of course we can still use molluscicide whenever that is relevant. There is an additional dimension here that is important. Look at these guys here in the picture. I mean, these prawns, the American, the African river prawn. It's huge and it's delicious, actually. And historically, this has been part of the diet of this population much through Western Africa on the coastline. This is a picture from Côte d'Ivoire, for instance, on the market. And so we started to think whether we could develop a bioeconomic model of integrated prawn aquaculture and disease control. And this is work that we've done with a former graduate student at UC Berkeley, with Justin Remains, who work also in our lab. Here we basically develop an aquaculture model in which you look at somatic growth, so how fast prawns grow, density-dependent mortality. We integrated them to have a function of total biomass on time, and we then use information of cost of management and the market value of the harvest every few months. And we identified that stocking prawns, for instance, and harvesting them every 6 months is an effective way to maximize profit. And when we pull this into an epidemiological model that accounts also for infected peoples and size structure and models from the snails, so that all the information that we acquire in the lab about the predator functional response over the snail, we can see that we can basically identify an approach that allow to keep control of schistosomiasis transmission in rice paddies and at the same time maximize the profit. We think this is so important that we develop with Tyler Higginson, who was a former master student in international cooperation of the Middlebury Institute for International Studies, and with Nicholas Joanar, we develop and analyze a business model. With Nicholas, actually, we built up a hatchery, a prawn hatchery in San Luis, at the border with Mauritania, and this is a gem in terms of aquaculture production in these areas that Nicholas is now running as an entrepreneur in this area. And now we are partnering through a Belmont Forum for Climate, Environment, and Health with David Little, Richard Nolman, Reed Ostrich and Rachel Cooney in Cote d'Ivoire. The first three are University of Stirling in Scotland, to continue to analyze how we can integrate aquaculture and health there for what we call the 4 pillars of wealth generating ecological solutions that are good because they increase food production, which is one of the sustainable development goals. They increase business to fight poverty, which is another sustainable development goal. They allow to recover ecological integrity, which is the sustainable goal number 14, and also address important public health problem, which is the sustainable goal number 3, with this idea that Sana Sokolov developed while we were working together in this area. We're looking, as a matter of fact, to an army of enemies, what we call enemies of my enemies. With Amberstein, we are looking at monosex Microbacterium rosenbergi. It's much easier to grow in aquaculture rosenbergi than Volenovi because there is a protocol for production for the Malaysian species, but we don't want to stock species that are not native to the environment, and using monosex individuals might allow to overcome some of the concerns of using only non-native species there. And we're looking also with Isabel Jones to other potential agent of biological control, including the tripod sterile grass carp. And I just want to mention that, for instance, it's not only about fish or aquatic species. The use of Indian ducks, for instance, in Asia, it's quite frequent to control the golden apple snails in rice paddies. And so this can also be used as a biological control agent. So in conclusion, finally, I just want to wrap up all that we have seen in this 3-part lecture. We've seen how precision medicine, as much as in precision medicine, there is an untapped potential for precision mapping of transmission risk for environmentally transmitted diseases, and this is certainly the case for schistosomiasis. We have seen that integrated ecological epidemiological models can contribute to optimize control of schistosomiasis transmission under limited resources, to understand what is the potentials. And of course, this should be come along also with all the necessary validation in terms of controlling for reinfection rates and prevalence of infection in people and in snail. And last but not least, something that I think is really important, we are disease ecologists and work in the area of epidemiology. It's really important to think in a systemic way to strive for win-win solutions that are able to target multiple sustainable goals at once, rather than thinking in silos and in compartment as like nutrition and health from infectious diseases and agricultural development and fighting poverty. We're different areas. I think they are all important, and we have opportunity to try to integrate them all together. If you have time, you can Google Protected by Prunes, and you'll find an 11-minute documentary that has been shot by the California Academy of Science of our work in Senegal, and really beautiful imagery, and I recommend you to look at that. And I conclude by thanking again the IACI 3D and the DAIDD 2021 crew for inviting me to be part and to, of this incredible venture school, and to share with you some of our research. Thank you very much for your attention, and do not hesitate to reach out to me by email or in one of our breakout sessions to talk about specific aspects of this problem. Thank you very much.